Alright, in this video I'm going to talk about the biosynthesis of this flavin molecule, this riboflavin, otherwise known as vitamin B2. And so riboflavin is made up of this isoalloxazine ring and D-ribitol, which comes from ribose, which is the sugar used in DNA. Uh, so deoxyribonucleic acid is ribose. And then when we make FMN or flavin mononucleotide, it's just adding this phosphate. And then flavin adenine dinucleotide, which is the molecule that riboflavin is needed for. It's the reason we need vitamin B2, and this is for used in redox reactions, and that's adding this adenosine onto it. That's why it's flavin adenine dinucleotide, and so we will go through here the steps in generating riboflavin, and I'll sort of quickly go through the two steps afterwards that make the flavin mononucleotide and flavin adenine dinucleotide, but those are pretty straightforward reactions, so I'm not going to spend as much time on those. Over here, this is just kind of to show where the ingredients for a lot of where the riboflavin come from. So we see up here, so glucose goes into the glycolysis right here, which, as I keep alluding to in videos, I will go through in very painstaking detail in later videos. But what we want to show here is that in the first step of glycolysis, you can go over into this pentose phosphate pathway, and that's how cells actually generate ribose, which is, as I said, needed for DNA and RNA. So through this pathway, we end up with ribulose 5-phosphate, which can then be shunted over into, over here in this orange or yellow box is the flavin biosynthesis. So we need an intermediate from the pentose phosphate pathway. And then over here, we see that we can get our GTP, which is our other starting ingredient, our guanosine triphosphate, and that can come from the purine pathway. And this is showing in well, essentially non-animal cells, because malate can come from this glyoxylate shunt, which, as I've written here, is an anaerobic alternative to the tricarboxylic acid cycle, which is the next step after glycolysis in sugar metabolism. It's also a step after fatty acid metabolism. So the TCA cycle is sort of a central hub for a lot of different biochemical pathways, particularly using this acetyl-CoA here. But this anaerobic alternative that some plants and fungi and bacteria can use is the glyoxylate shunt, and that can generate malate here, which then goes to biosynthesizing GTP in the purine pathway, and then GTP can actually be used as the other starting material here. Down here, I just put this in here because in the mechanisms that I show, I'm actually using sort of the long form names for a lot of these enzymes, but they have these short form names. So the GTP cyclohydrolase is RibA, Rib for riboflavin. The pyrimidine, deaminase, and reductase steps are done with, well, there are several different Rib enzymes that can do this. But the one we will be looking at is RibD. Then there's this, what they call just broad spectrum hydrolases. So these hydrolases are phosphatases. So phosphatases, so they are just removing a single phosphate. And the enzymes doing this in this pathway were actually unknown until quite recently, like within the past decade, really. Uh, there's no structures for them yet, as far as I could find, but the actual phosphatases used have actually been identified. There are a few of them that are possible. Then over here we have RibD, which is the 3,4-DHBP synthase, which is what's taking that ribulose 5-phosphate out of the pentose phosphate pathway. And so this is one of our starting materials, and this is our other starting materials. And then this lumazine synthase, or RibH, comes in and combines those together into this intermediate here, and then riboflavin synthase, or RibE, turns it into riboflavin. And over here, this is just a bunch of the proteins that can actually bring riboflavin from the diet, so dietary riboflavin, into the cell. So there are a number of different receptors here that can do that. So down here is just a little bit more detail on this pathway. So we see, once again, first we start with this GTP, then this GTP cyclohydrolase, releasing this this molecule here, is opening up this ring right here, and so that it's just opening that up right here, so that we end up with this single ring. And then there's this sort of interesting little thing here, where in bacteria, in plants, there is a single protein, a bifunctional protein, that does this in the order of the deamination, so removing this amine and turning it into a ketone. And then this reductase step here, which is actually opening up the ribose sugar ring into this linear carbohydrate chain here. However, in fungi and archaea, there's two different enzymes that do this, and they do it in the opposite order. So they do the reductase first, opening up that ribose ring using this RIB7 enzyme here, this pyrimidine reductase, and then this RIB2. I think in some species it's actually RIB3. It can be either of those, depending on the species. And then, the deaminase step actually happens afterwards. So once again, removing this amine and turning it into a ketone. And then this step here is that one that I was talking about, which was only recently discovered. So they don't even have like a fun name for it here. It's just these kind of groups of letters here. And so there's the ATCPFHY/PYRP1, or the ATPYRP2 here. And that generates this species right here, which, as I said, is just removing this phosphate. That's all it's doing. And then there's this weird thing going on here, which we'll get into in more detail later, where there's this cyclic pathway here. But this is where that carbon chain from the pentose phosphate pathway actually comes in. So the ribulose 5-phosphate is broken. A carbon is taken off, so that we go from a 6 carbon to a 1, 2, 3, or 5 carbon to a 4 carbon chain here, and then that gets added onto this right here, so that we end up now with this ring on here. And then this riboflavin synthase is kind of an interesting step because it ends up generating this sort of large, like 5-ring intermediate, but then one of the rings is broken, and the 3-ring product that comes off is the riboflavin, and the 1-ring product that comes off, because, like I said, we're breaking a ring off, or we're kind of breaking a ring in half. It's like we have 5 different rings on it, and one of them is getting broken in half, so we end up with a 3-ring structure and a 1-ring structure, which gets recycled back to this other step here, where the lumazine synthase then comes in, bringing this 4-carbon carbohydrate here into it, and sort of repeating it in this cyclic pathway here. Then the riboflavin, so this is actually the dietary molecule here, but sort of what happens to that is that it gets turned into the form that's actually used for redox reactions. And the first step is adding this phosphate back on, which might seem kind of almost a little counterintuitive. We have this step here that takes it off, only to just add it back on again later. But as I've said in previous videos, evolution is a blind watchmaker, so it doesn't look at this and go, well, I could just simplify this. However, it's also possible that that this is a better way to do it, because the phosphates kind of they come off somewhat easily, which is sort of the purpose that they serve, is that it's energetically favorable for them to get removed from molecules. So if we had this as the dietary form of the molecule, then it might be that the phosphates would sort of come off, and that the molecule as a whole, with the phosphates coming off, would degrade rather quickly. But anyway, the next step is then this FAD synthetase, or flavin mononucleotide adenylyl transferase, which is actually adding an AMP onto the end of it here. So it's just adding the AMP, and so the pyrophosphate coming off of ATP is released, and we're just adding this this AMP onto it. All right, so let's get into more detail here. So the first reaction, like I said, is the GTP cyclohydrolase 2, and it's called 2, meaning obviously that there is a 1 version, and the 1 version is actually used by bacteria for making folate, and in animals it's used to make a molecule called tetrahydrobioterin, which is used as a cofactor in neurotransmitter synthesis. And humans actually make that, which is why we don't need that as a vitamin, which is why we don't need the tetrahydrobioterin as or biopterin, you might pronounce it. But anyway, the reason we don't need that in our diet is because we actually make it, but we do need riboflavin in our diet. So this GTP cyclohydrolase 2 is the first step in making riboflavin. And so the first thing that happens is the GTP here. So this molecule here is the guanosine triphosphate, goes into the active site, and this arginine 128 actually binds to the first phosphate on it, so it releases pyrophosphate. So the pyrophosphate comes off as we form this covalent intermediate. And then, so there's this water here that's coordinated with this zinc in the active site, and this tyrosine 105 actually uses its hydrogen here as an acidic hydrogen. It's donating that hydrogen to this double bond here, so that we are adding this this hydrogen on here while the water is also attacking onto this, and we end up with this hydroxyl on here, which is still coordinated with the zinc. Uh, and so then the next step, we move down here, and another water actually in the active site is now attacking this carbonyl carbon here, which is still coordinated with zinc. I guess I could have specified up here that this hydrogen is being released, and we're collapsing this back down into a double bond to create the ketone. And then this water attacks this ketone here, and this can actually break the bond here, while also releasing formate. So this formate is made out of the carbon and oxygen here, and it is breaking this ring open here. And so we can look at the structure of it here, and we have the well, this is an analog of the GTP. We can see it actually has this extra carbon in here, and that a lot of times crystal structures are done with analogs like that, so you can actually see what the substrate looks like in there, so that this pyrophosphate here does not get hydrolyzed off of it. We can actually see how the substrate is sitting in the active site. And so we can look over here. So this in blue is that zinc, and then this little sphere right here is the water molecule. And so we can see that this is coordinated. Might be hard to see, but there's this, it's 3.1 angstroms away from that zinc, and 3 angstroms away from this carbon that it will be attacking, and 2.4 angstroms away from this, from the tyrosine that we saw here, this tyrosine 105. And so we can see how that water is brought close to this carbon here, so that it can attack that. And then the authors suggested that this water down here, this little red sphere, is the second water that's being used down here to attack that carbonyl carbon right there. I don't know. That seems maybe a little far-fetched to me. It's quite far away from this. I guess in later steps it could be brought closer, but I don't know. It's possible, but this water up here definitely, that definitely seems to be likely. It's sort of sitting right in the exact spot that it ought to be. And so I also have here, you can see these cysteines that are in the purplish color holding that zinc in there. We have a magnesium coordinating with our phosphates up here, which is pretty common for phosphates to be sitting there with a magnesium. We have up here, so this is an arginine, and that would be the arginine 128 here. That if this didn't have that carbon in the middle, if this were a phosphate, would have bound to this phosphate here and kicked the rest of this off, creating that covalent intermediate. Yeah, and so that is the structure and mechanism of the GTP cyclohydrolase 2. So now this RibG or RibD or the Rib7 and Rib2, so the yeast pathway doing the reduction and then the deamination first, the bacterial pathway doing the deamination, the reduction first. And so they've broken this up. So this is actually in RibD. I could have added RibD up here as well. and in fact, RibD is the structure that I'm going to be looking at, so I probably should have put that in there more prominently. But this is the RibG, the RibG deaminase, so D for deaminase mechanism right here. And so it's using this E51, this glutamate 51, which is, so we're once again using water here. It's deprotonating the water, which is coordinated to a zinc once again, and then that water will attack this double bond here and kick off this this amine group here, and then the oxygen from that water is what's used to generate our ketone there. And so then the reduction step is is actually going to use, well, it's using another water right here. So this water, so this shows the water actually deprotonating this nitrogen here. It's possible that it could be this glutamate 290 deprotonating that. So it's the point is that this nitrogen here is being deprotonated, and so the electrons are jumping down there to create a double bond there, and then the electrons from this bond here are going and grabbing that hydrogen back off of the glutamate 290, and it's breaking this bond right here, and so it's linearizing the ribose sugar there, and then it uses NADPH, which is a molecule used in redox. It's pretty much the same as NADH, nicotinamide adenine dinucleotide, but it just has that extra phosphate on it. But anyway, it's donating this hydride right here, which is a hydrogen with 2 electrons and a negative charge, is going down on here. So it's going to attack that carbon right there and turn it into an sp3 carbon, so a carbon with just 4 single bonds. And then the electrons from this double bond here are going to go and grab an H+, so a hydrogen without any electrons. And so we end up with the carbon with this hydrogen on it. And this carbon should actually have another hydrogen on it. It's sp3, and this nitrogen now has the hydrogen that was donated here onto it, and we end up with our product there. So down here is a multiple sequence alignment for the RibG, the RibD, and the Rib7, which is, as we see over here, the Rib7 is the reductase step for fungi in archaea. And so we can see that there are quite a few conserved residues. So everything within boxes are conserved residues. So the RibG and RibD are quite conserved. We don't start getting the Rib7 until over here, because the Rib7 does not contain the deaminase step that RibG and RibD do, which we see here is the first step, and the deaminase step is actually in the N-terminal domain of the proteins, and so it's at the beginning of the proteins. And then we see the reductase domain of the RibG and RibD starting here. And we see that, I mean, there are quite a few stretches here where there isn't that much conservation, but then we see some stretches here with quite a bit of conservation between all 3 of these enzymes. And so I have the structure of them here, and this is the RibG. The RibD I can add in here, and that's in green. So we can see that contains up here what's in white is the deaminase, the deaminase parts of the enzyme there. Uh, and so the green also contains that, and then the purple is the reductase part. We can see that the green, when I add that in, overlaps with both of those. It just doesn't dimerize, or actually it's this is a dimer, so it's not actually making this tetramer of it here with the RibD. Then I can add the Rib7 in red, and we see that that is only in the reductase part of the RibD there. And so we can look at the different active sites here. So over here is the deaminase active site. And we can see our substrate in here. We know it's substrate because we see it still has this amine group on it. And this has a zinc ion in here. And we know from the mechanism up here that we have the glutamate 51, which we can see, where is that glutamate? So that would be, well, I think I have this in the other part of the dimer here. So that's that glutamate, but we can find that here. That would be that glutamate right there. So yeah, that's the glutamate 51 in this active site. And we can see that that's brought in close to this ring here, which we see is similar here in our mechanism with that zinc ion there. And so we can see, I don't see, oh, it might, maybe you could say that this water, I don't know, that's a little bit far away. Trying to see, maybe this water down here is the water they think is attacking that carbon there and replacing the amine. But anyway, it's likely that water right there. Although it seems like it would be closer to the amine here. Perhaps, oops. Perhaps this water right here, this little sort of asterisk-looking thing, is. So I could maybe make that a little bit bigger so it's easier to see. Make it a nice big sphere. Oops, I still had that other thing highlighted. So hide spheres. So let's just highlight this water, oops, this water here, and we'll show that as a sphere. And so you can see it a little bit easier. That's still, it looks pretty far away from everything, but anyway, I'm not going to belabor this point. We can see that this glutamate is brought in close, and that this is coordinating with the zinc ion here. And then we can move on over to our reduction step here. And by the way, the paper, or at least one of the papers that I was looking at, said that they do not think that there is a tunneling of the substrate going on here, because this step happens faster than the second step, which means that you would be releasing this out of the active site sort of into solution, because this step here wouldn't be able to keep up with the first step. And so they do not think there is substrate channeling going on. And so this here is the reduction step. So up here is, you can see our ribose is now linearized. Up here, we still have our single ring there. And down here we have our NADPH, so it's bringing that nicotinamide moiety up here into the active site. And we see that that is brought close here where it's going to it's going to reduce, well, it would reduce the cyclic ribose here. So this is actually the product of that. So we know from our mechanism here that it's breaking this, it's breaking the carbon to oxygen, this ether bond here on the carbon oxygen to linearize that. Oops, wrong one. Okay, so so it's yeah, it would be breaking this oxygen off of there, because we see here this makes a 1, 2, 3, 4, 5 member ring. So this carbon here to this oxygen here is the bond that is being broken. And this mechanism said that there's this glutamate 290 here. So that would be the glutamate 290 would be likely maybe this one here. So let's take a look at that. Yep, that's the glutamate 290 right there. So we see that that is brought close to the ring that it's going to be breaking as well. Yeah, so anyway, that is the the enzyme, the bifunctional enzyme here, where I have, as once again, I have the reduction part, the C-terminal domain, that's the reduction domain in this sort of purplish color, and the deamination part in this white color. And as I showed before, the reduction the reduction enzyme for the fungi and archaea overlaps pretty well with the reduction domain of plants and bacteria. All right, let's move on to the next step here. And so this is that that dephosphorylation step that I was talking about, where it was only recently discovered using these kinetic assays here. They looked at the activity of the two enzymes that they isolated as being able to do this phosphorylation step. And we can see here that the Km of the binding. So it looks like the ATPYRP2 is not binding the flavin mononucleotide, which is being used for this. Well, actually, no, it's not being used. This is just a dephosphorylation. So they do see that they were looking at whether FM and N can be dephosphorylated, because as we saw in the pathway up here, FMN is just a phosphorylated version of riboflavin. So they were looking at whether the FMN can be dephosphorylated by this, and it looks like with the first enzyme there is activity against the FMN, and in fact it's binding tighter, but its turnover rate is worse. So the ARPP, which is this 5-amino 6-ribotylamino 2,4,1H3H pyrimidine dione 5' phosphate, which is what the ARPP is short for, is binding well, about an order of magnitude less binding affinity than the FMN, but it's about 3 orders of magnitude better at turnover rate. And so, yeah, this one seems to be able to dephosphorylate both of them. So I couldn't find a structure for any of these. Uh, they did mention that these are members of the haloacid dehalogenase, or HAD superfamily, which has it has a pretty conserved sort of mechanism. Um, I don't think I put that mechanism here, uh, but yeah, they it's I mean they call it a superfamily because there are a lot of enzymes that use this same sort of mechanism, and they're called haloacid dehalogenases because a lot of them actually take halogens, which are things like chlorine and bromine and iodine, and I guess fluorine off, particularly things like chlorine. So removing chlorines from hydrocarbons. But the same mechanism is used for removing phosphates as well. And so it uses, so the enzymes they found were members of that HAD, that haloacid dehalogenase superfamily. So the next step I have up here is actually this DHBP synthase. So as we saw earlier, that's the one that starts from the ribulose 5-phosphate, so from the pentose phosphate pathway. And so this uses this mechanism here, so this aspartate 185 actually deprotonates this carbon here, the third carbon in this 6-carbon chain. And so this hydrogen is removed, the electrons from it collapse down, these electrons move up to the oxygen. So we have this enediol intermediate here, and these oxygens are being are coordinating to these metals here to stabilize them. And I have this over here because this is a similar mechanism, this enediol formation to rubisco, this ribulose 1,5 bisphosphate carboxylase oxygenase. So this is a similar species to what's starting out over here. This is ribulose 5-phosphate. This is ribulose 1,5 bisphosphate. And so the only difference is that this oxygen here is phosphorylated on it. But it's doing the same thing where it's deprotonating this third carbon, which is generating this enediol here. And then in rubisco, it can either add carbon dioxide or oxygen, where this is the not favored reaction here. This is sort of a side reaction, which requires a bunch of steps to sort of turn it back into something usable. But this is the carbon fixation that plants do. And the reason this is interesting is because rubisco is an incredibly ancient enzyme, and it's also the most abundant enzyme on the planet Earth. It's the enzyme that plants use to actually take the carbon dioxide that they breathe from the atmosphere and affix that to this ribulose bisphosphate here, this ribulose 1,5 bisphosphate, and turn it into these 3-carbon species here, which can then go on to become sugar. And so that's an extremely important but somewhat irrelevant, I guess, here. But I just thought it was interesting that this uses the sort of the same mechanism here. But anyway, we generate this enediol, but then the electrons from this oxygen here collapse down. The electrons from this double bond here move over here, which kicks off the OH here. So we end up with this carbon double bond to carbon, and the H2O coming off here was from that hydroxyl group there. And then we actually do we actually have the electrons from this collapse down. These electrons here, I guess you could have come and grab that hydrogen off of there, so we end up with this double ketone species here. And then we actually have these electrons collapse down here, and the electrons from this move over here, and the electrons on this double bond move up. And from that we end up here with this two ketone species here, where now instead of the ketones being adjacent to each other, they're one carbon away. Once again, everything's still coordinated to these metal ions. Then we have an H2O come in and attack this ketone carbon here, which so a lot of times in these diagrams you'll see these squiggly lines, and that's showing that the stereochemistry of these bonds is not determined. These could be the OH could be down here and the H could be up here, or it could be the way it's shown. It's not really relevant, I guess, for this intermediate which way that the stereochemistry is done. And so this is actually adding another H2O. So I just kind of drew it in here so I could show that it's attacking this this carbonyl carbon right here, or this aldehyde actually. I mean, it's a carbonyl, but it's also an aldehyde. And so we end up with that water right here as this OH. And then we have the electrons collapse back down here, and then the electrons from this move over here. The electrons in this double bond move up onto the oxygen, and so we are actually releasing formic acid here. And so the formic acid is actually coming from this oxygen here and that carbon right there. So this moiety right here is actually being released as the formic acid, and so we end up with this 4-carbon species here. And so this is then released. Well, it actually goes through another enediol transformation where these electrons move down here, and then these the electrons in this from this double bond can then pick up another hydrogen. That's why it's showing this plus H plus here, and then we end up with our DHBP right here. And so I have the structure for that right here. So we can see our two metal ions here, which they are saying is zinc and calcium. So zinc is this darker sphere here, and calcium is this larger one right here, which, yeah, looking at the periodic table I have, they're both in the same row, but zinc is in a later column. So I guess it makes sense that it would be smaller than the calcium, because adding more protons tends to shrink ions down. And so we see down here, if we look back at our aspartate 185 here, is this aspartate right here. And so I have the measurement there, 3.5 angstroms from this third carbon. So we can see how that could be deprotonating that carbon. So in the rubisco, that's actually happening here with this carboxylated lysine. So we have our lysine right here, and then this carboxyl group is being added to it. And that's why I have this picture here. I didn't want to look at the structure as a whole, because this is kind of a side thing here. But we see this carboxylated lysine. We see that's about 3.1 angstroms from that third carbon there, which is close to what we have going on here in our DHBP synthase. Yeah, and so we can see the two metal ions there. We can see our aspartate 185 right there. That is pretty much mostly what I wanted to show here on this structure. We do see that this is in a dimer form, and some of the amino acids in the active site here are actually being brought in from the dimer, and so interestingly, this serine here looks like it's modeled in away from the cartoon ribbon there. Well, one other thing too, I think the paper mentioned that this loop right here undergoes a conformational change when the metal ions actually coordinate into the active site. And so we can see that this arginine here is this arginine and this glutamate here are both sort of going into the active site here to coordinate with those metal ions. All right, so now let's go back here just to kind of get our bearings a little bit. So we've now gone through these first steps here from the GTP or GTP cyclohydrolase, broke this ring here on the GTP. And then we could go through this deaminase reductase step, which is reversed between fungi, archaea, and the bacteria in plants. And then we had this dephosphorylation, and we also took this ribulose 5-phosphate and generated this 4-carbon species here, this 3,4-dihydroxy-2-butanone 4-phosphate, shortened to DHBP. And so now this lumazine synthase is going to bring these together. So I'll go through the mechanism first before I get to those interesting structural stuff about this enzyme. So this is our the product that we got from the GTP, and this is our DHBP. And so this amine group here is actually going to attack the carbonyl carbon on DHBP. We're going to form this shift base here, and then we're going to deprotonate this carbon right here, and the electrons will collapse down and kick off the phosphate here, generating this double bond right here. This shows that this just sort of rotates around that single bond there, bringing this carbonyl right here in close to this nitrogen right here. So it's actually kind of skipping another step, which would be the electrons from this collapsing down, and the electrons from this, I guess we could have go and grab that hydrogen off of there to regenerate our ketone here. And then we have this nitrogen here attacking that carbonyl carbon, which causes these electrons from the double bond to go over and grab a hydrogen ion right there, which now forms this 6-member ring here. And then this shows that this nitrogen here is being deprotonated. The electrons collapsing down here, the electrons from this double bond moving over here, this double bond moving over here, and that is then kicking off this OH here, which they show grabbing this hydrogen right here to create water, and now we have this 2-ring system here. And so this I mean, this is a fairly straightforward mechanism. It's just a lot of carbonyl chemistry, which is quite common in biochemistry. But what's really interesting about this enzyme is that it forms this large structure. So it's this monomer is a pretty small protein, but they come together in these pentamers, which then form into these decamers, which we can see from a side view and a top view. Uh, then these decamers actually come together into this large 1 megadalton dodecahedron of 12 pentamers. And it's this large sort of hollow structure, this dodecahedron. So I have, so this is actually the pentamer right here. And before I get too far into that structural stuff, let's just look at the active site here, which is actually between two of the monomers here. So residues are coming in from two different monomers. And we see this structure here, which is actually an analog for the substrate here. And we can see the well, I don't think I went into too much detail here with what the residues are, but we can see that how that fits into the active site, which is at the interface between two of the monomers. And so, yeah, like I said, that is this is a oops, this is a pentamer right here. And then we can actually add in, so I think I have that on here. Where do I have that? Just a second. I thought I had larger structures going on in here. So I think, yeah, I think if I do this, yeah, so this is several of those pentamers. This is 4 of those pentamers coming together into a single structure here, at least part of a structure. And then in the next structure, I actually have the full dodecahedron right here. So this is a bunch of those pentamers brought together into the dodecahedron. So I highlighted a few residues here, which will become a little bit more relevant as I talk about something else that they have. Well, I don't think I have pictures of it, but I do have the structures of it. And in this paper, they actually did some site-directed mutagenesis, and that is they did it on these residues I have in green. So this is just on a single monomer here. They did this, that would be present on all of the monomers in here, which I think there are 60 monomers on here. And so when they mutated those 4 residues, which is an arginine 83 to glutamate, a threonine 86 to glutamate, a threonine 120 to glutamate, and a glutamine 123 to glutamate. It actually caused it to generate this this much larger structure, which I have shown here in sort of this rubyish color. I think I'm going to change the color on that so that it's easier to see. So yeah, I change it to blue, so you can see that it creates this much larger structure here. So it causes a slight change in the way that the different that these different pentamers actually associate with each other. It creates kind of a different angle between the different pentamers together and creates this much larger structure. And what's interesting, so I guess to kind of skip ahead a little bit on here, the next step of this reaction is the riboflavin synthase. And as I show here, this actually sits inside of the dodecahedron of the lumazine synthase. So it's encapsulated in that dodecahedron, which we can see here in the magenta color. When they mutated those 4 residues to a glutamate, this larger structure was actually able to encapsulate other proteins, particularly proteins that they added these 10 arginine tags onto. Arginine being positively charged, the residues that they mutated being negatively charged. So it actually encapsulates those positively charged proteins. But then they actually went through and did, instead of site-directed mutagenesis, they did what's called directed evolution, which is essentially just generating mutants using what is called so it's a form of PCR that doesn't have good fidelity, so it can introduce different mutations pretty easily. And so they did that and introduced several other mutations and got this even larger structure here, which you can see. You can see the natural dodecahedron sitting here. Then in blue is the larger structure, which they called the LS negative, because they introduced those negative residues in it. And then this larger one they called the LS13, because it had, in addition to those 4 glutamate residues that they introduced, it had several other mutations, giving it 13 different mutations. And so we can see that it generates this large spherical structure here. And you can actually zoom in right here, for instance, and see one of those pentamers here generating this large spherical structure. But anyway, I just thought that was really cool that just through a little directed evolution, they could have it generate this large structure. It looks pretty large, but if you think, I think it's something like 3 megadaltons. But if you think about like a virus structure, which is a giant protein structure, those can be like in the range of thousands of megadaltons. So, but you can see how a large structure like this can sort of come about through evolution. But let's move on to the next step, which is the riboflavin synthase, which, as I said, actually sits inside of the, so if I get rid of these structures, it actually sits inside of this dodecahedron structure. So we can actually zoom in until we're sort of inside this dodecahedron here. And those would sit inside of it. And these pores here would actually be large enough that the riboflavin that's generated on the inside could then move out of the structure. And so the riboflavin synthase has several proposed mechanisms here. I'm not going to go through mechanism A because it's known that this is not the right mechanism, because it doesn't go through this intermediate, which has been discovered to be a known intermediate, which is this 5 cycles, this pentacyclic structure here. And so one of the proposed mechanisms is this one here at B. So we start out with these 2 ring structures here, which, as I said, is because of this cycle going on here. So this single ring structure here through the lumazine synthase has this added on, so that we have the double ring structure here. And so we have a new one here that that was generated from this this lumazine synthase here. But then we also have uh, well, we actually start with two of these single ring structures because one of them is left over after the riboflavin synthase reaction, and we won't see that here. So this one shows that two of the double ring structures actually come together, and this double bond here, this ethylene moiety here, attacks this double bond here, which then goes and grabs a hydrogen ion. So now we have both of these bound together right here. So this carbon here is the same as this carbon here, and it's now bound to that carbon right there. And so then we actually have, we actually deprotonate this right here. And so this generates this double bond right here. And so the double bond there can then sort of move, well, this double bond here is actually going to move onto this right here, generating this other ethylene here. And then that can actually attack this carbon right here, sort of doing the exact same thing we did up here, but now we are forming this, this pentacyclic ring. But another paper actually proposed this other pathway here that that once again it's bringing these together right here, but this actually shows what their mechanism proposed is this hydride shift. So this hydrogen here bringing two electrons negatively charged, going down here and causing this double bond to sort of move over here, this one to move over here. This double bond goes and grabs this hydrogen ion here, this hydrogen cation. And so this hydride shift here generated this, this double bond here. And so now we have these two double bonds, these two conjugated double bonds. And this is what's called a diene, because we have these two ethylenes here. And so this over here, well, I guess through so this hydrogen would get added onto this nitrogen right here, and this double bond would move over there. But what they were trying to go for is that we end up with this type of thing right here, where we have this diene and this, which is what's called a dienophile, meaning essentially that it loves dienes. And then it can go through what's called a Diels-Alder reaction, which I have over here. I won't go too much into the molecular orbital theory of it, but just know that that there is this highest occupied molecular orbital and this lowest unoccupied molecular orbital, and the phases of them are such that that they can be brought together such that the phases overlap. So this up here is our diene. There is double bonds going on right here and right here, and this is a double bond right here. And so this it's the lowest unoccupied molecular orbital. They can overlap with the highest occupied molecular orbital of the diene, and they are in phase. That's why we see the darker one here overlapping with the darker one there, and the lighter one here overlapping with the lighter one there. And this actually, so bringing the diene and the dienophile together, you can generate a ring like that. And so often the mechanism for that is shown like this. It's not actually happening like this, but it's a good sort of bookkeeping method. Having an arrow go here, this arrow go here, this arrow go here. And so we end up with this ring structure here, which has that single double bond, which is just like this ring structure here with that single double bond. and then that can also generate our known intermediate here. So this B pathway here and this C pathway here are the two proposed mechanisms for the riboflavin synthase. But as I said, this pentacyclic structure here is a known intermediate. So this one they know has to be part of the mechanism here. And so these two, B and C, are mechanisms that show how you can get to it. This A mechanism was proposed before this intermediate was known to be present in this mechanism. But then we go through this ring breaking structure here. So this double bond moves here, this double bond moves here, this one moves here. We are breaking this bond right here, which is going and grabbing onto a hydrogen ion. We're doing something similar here. It looks like they are deprotonating this hydrogen right here. The electrons move down to generate the aromatic ring here. These electrons break off and move up here, so we are breaking this bond here and this bond here. So we end up with this single ring structure and this triple ring structure, and this right here is riboflavin. This R here would just be that hydrocarbon chain made from the ribose sugar. And then this single ring structure here once again goes through the lumazine synthase step. So the single ring structure coming back here. We have a riboflavin coming out here, the single ring structure going back to the lumazine synthase. This structure, this 4-carbon hydrocarbon chain from the pentose phosphate pathway is added to it from the lumazine synthase, and then that goes back up to replenish our double ring structures used here. And so that is the cyclic pathway part of that. And then so the riboflavin, which is kind of what I was trying to get to. Well, let's actually take a look at the so this is the riboflavin synthase. I have each of the 3 subunits in sort of a different greenish color. So we can look at the structure up here. They have this. They have the 3 of them colored in different colors here, and the active sites are actually in the interfaces between the 3 monomers here. And so we can zoom in. So it's actually an asymmetrical structure. So we see here that 2 of these subunits are brought together where these ones are split apart. And so what seems to happen is that these subunits are mobile. They can move around and bring interfaces together when substrate binds and then open up on another one to release product. And so we can see here this is 2 of these brought together. So these are 2 of these substrate analogs brought together here. So we know they're analogs because these are just in orange here are single ring structures, but we know that our substrates in the actual protein are double ring structures. And so we can see that one of them is brought using this monomer right here, and the other one is brought in using this other one. And so the 2 are brought together at the monomer interface there. And we can look over here at the open conformation, and we can see that this lysine here is sort of sticking out, whereas if we go over here, it's now sort of that is a lysine, right? Yeah, that's a lysine. And the lysine over here is actually being brought into the active site right here. And so we can see this that there's this large conformational change when those things come together. Um, I think, uh, yeah, yeah. So we have them sort of brought together in that one over there. Yeah. So yeah, you can see how this is sort of a dynamic enzyme where these subunits would constantly be sort of wiggling back and forth, bringing substrates together and then opening up to release product from it. Oops. Um, and so that's the riboflavin synthase. Then the next steps after that to turn it into flavin adenine dinucleotide is, as I said, just this, um, just adding the phosphate through this riboflavin kinase, and then this flavin mononucleotide adenylyl transferase is just adding this AMP onto the end of that. And I do have the structures for that. I'm not going to spend too much time on them. This is the riboflavin kinase. We can see our flavin adenine dinucleotide here, and we can see this ADP here. So this is the product here, and this has already released the phosphate from the ATP. But we can see how they're brought close together, so this phosphate could easily be transferred from here onto here. So onto the flavin mononucleotide right there. And then we have over here, this is the FAD synthase or the FMN adenylyl transferase. And we can see this has our substrate here, which is our flavin mononucleotide. And then we have this ATP analog. Once again, it has this carbon between the two phosphates here so that they don't hydrolyze. So we can look at the substrates in the binding pocket in the active site here. But we can see how this right here could be brought in and bind onto this phosphate here, releasing pyrophosphate and adding this adenosine onto the end of this FMN here. And we see that this has 3 subunits here. And yeah, once again, it has a magnesium ion in here, very common for phosphate stabilization. But yeah, that is how you go from riboflavin, which, as I said, riboflavin is the form of vitamin B2. This is the dietary form. This stuff happens after it is ingested, so it's not technically part of riboflavin biosynthesis, but it is important to generate this this active form here, which, as I said, is used in redox reactions. All right, so let's just sort of quickly sum up what we've gone through here. So we started with our GTP cyclohydrolase. We removed this formate here to generate this intermediate here where the ring is broken. We can then either go through the reductase deaminase or the deaminase reductase, depending on whether it's in fungi and archaea or if it's in bacteria and plants. Then we have this dephosphorylation step where the enzymes involved in that have only been recently discovered. I think the paper for it might have actually been from 2016, if I'm remembering correctly. But anyway, that dephosphorylates it. Then we go through this cycle here of the lumazine synthase, and which generates this double ring structure here. Then the riboflavin synthase brings two of these together, so times two, to generate riboflavin and also kick off one of these single ring structures here. And then I talked about a little bit the riboflavin kinase and the FAD synthetase, or as you actually find it on the protein data bank, the FMN, or flavin mononucleotide adenylyl transferase. So yeah, and so that is riboflavin biosynthesis. I hope you found this video to be enlightening and informative. In the next video, we'll be talking about vitamin B biosynthesis, which is niacin, and I will see you in that video.